We recently presented at the CIMT meeting at Mainz our new target that came from our computational discovery platform of Compugen. And this is another program we identified, which is actually harnessing cytokines to treat cancer in a very different way. Now, we all know that cytokines are very powerful tools and immune-stimulatory, but they're not very good drugs because eventually you give them peripherally and they stimulate the periphery and many times induce toxicity. And then the challenge of therapeutic window is how can you get sufficient amount of cytokine in the tumor microenvironment to actually induce anti-cancer immunity without reaching first a peripheral toxicity. And this is a challenge of cytokines, and while there are approved cytokines, the last 30 years, people are trying by different ways to pegylate, to engineer, until this is a major challenge for cytokines. And what we identified is a target which is called IL-18 binding protein, which is inhibiting naturally IL-18 in a tumor microenvironment. So the data we presented have shown that in the tumor microenvironment of many tumor indications, you have quite a lot of this potent effector cytokines, which is called IL-18, and it's floating there in an inactive form because it's being bound by IL-18 binding protein. And what is also very important is that IL-18 is quite low peripherally. So you have high levels of this cytokine, not active in tumor microenvironment. And then our approach, and we are the first to show that, are using an antibody as for other checkpoints, which is a drug moiety with all the advantages of a drug. And we unleash and break the interaction with IL-18 binding protein and IL-18, and by that we unleash the natural activity of IL-18 to be active in tumor microenvironment. And because that the pathway of IL-18 is really most dominant in the tumor microenvironment because it's induced by inflammasome and by inflammatory signals in tumor microenvironment and low in the periphery, actually our approach overcome many of the challenges that I've seen for other cytokines because we are not a cytokine program. We are releasing a natural cytokine which is upregulated in the tumor microenvironment to really modulate and increase anti-cancer immunity without modulating the periphery. And by that, we are very different from all the cytokines and all the other approaches. Now, in general, there is I would say, renaissance of IL-18. There are different now developments of IL-18, but this is the way we are doing it, which is not using a recombinant cytokine, but actually the drug is very different. And what we have shown in CIMT meeting is that we see very potent monotherapy activity when you block IL-18 binding protein in human systems, in mouse settings, tumor models. And while the tumor environment after treatment is completely immune modulated, the periphery is completely clean. And again, if you look at every cytokine development, you see this peripheral increase in T cell proliferation, cytokines. Our periphery, at least in the mice and also in some human systems, is completely clean, and only the tumor environment is being modulated. And by that, our approach is unique and supposed to bring a safe, efficacious cytokine approach to treat cancer patients.